Hello everyone. Today our topic is transcription unit. So what is transcription unit? Transcription is the part of the central dogma, and transcription meaning production of RNA from DNA. Okay, or simply you can say copying of genetic information from one strand of DNA to RNA is called as the transcription process. And now what is transcription unit? So, transcription unit meaning that part of the DNA from which the RNA strand is formed. Okay, that part of the DNA or that nucleotide sequences of the DNA from which the RNA strand is formed, that part of DNA is called as the transcription unit. Okay, so here say. So transcription unit, the segment of DNA that takes part in transcription, that is called transcription unit. Okay, the segment of DNA, not the whole length of the DNA, only few specific sequences of that DNA molecule take part in the process of transcription, and that specific region of DNA molecule is called transcription unit. Okay, so this transcription unit consists of three components. One is called promoter, structural gene, and terminator. Remember, the transcription unit consists of three components: promoter, structural gene, and terminator. Okay, so now let's move to the next slide. So first component here, say the structural gene. So now what is structural gene? So structural gene meaning the DNA strand which polarity is from 3' to 5' direction. That strand is called as the structural gene. Here say, it is the component of that strand of DNA which has 3' to 5' polarity. So DNA is double stranded. Okay, that DNA strand from which the single stranded RNA is formed, that strand is called as the structural gene, and that structural gene has 3' to 5' polarity. That means the direction of the strand is from 3' to 5' direction. Okay, this is called polarity. Okay, so remember, from only one strand of DNA, the single stranded RNA is formed, and that single or one strand of DNA is called structural gene, whose direction is 3' to 5'. Okay, I will show you later in the diagram. Okay, now see next. Yes, see, this strand of DNA is called as the template strand or antisense strand. Remember these highlighted names: template, antisense. That means that DNA strand from which the RNA is produced, that strand of DNA is called template strand or antisense strand, and the opposite one, which polarity is from 5' to 3' direction, that strand is called as the sense strand or coding strand. Remember, the sense strand or coding strand does not involve in the process of the transcription process. This is very important, guys. Please do remember, the strand of DNA from which the RNA is formed, that is called template strand or antisense strand, whose direction is 3' to 5'. Okay, the opposite strand is called as the coding strand or sense strand, whose polarity or direction is from 5' to 3' direction. But this strand of DNA does not involve in the process of transcription. So what is the difference between the replication process and transcription process? In case of replication, both strands of DNA serve as the template strand from which two new DNA strands are produced. But in case of transcription process, only one strand of DNA is involved, from which the single stranded RNA is formed. Okay, and that strand of DNA is called template strand or antisense strand. And remember, the polarity of the template strand is from 3 prime to 5 prime direction. Okay, the polarity means simply the direction of the strand is from 3 prime to 5 prime. Only from this strand the RNA can be synthesized, and the newly synthesized RNA strand's direction will be from 5 prime to 3 prime. Always remember, any new strand can be formed from the 5 prime to 3 prime direction. Why? Why always the new strands are formed from 5 prime to 3 prime direction? Because the 3 prime and always contain one hydroxyl group, OH group, and the DNA polymerase enzyme, RNA polymerase enzyme, all this enzyme binds the nucleotide triphosphate or nucleoside triphosphate to this 3 prime end of the strand, and that is why always the strand elongates from 5 prime to 3 prime direction. Okay, so this is the direction of the newly synthesized RNA, which will be 5 prime to 3 prime. Okay, and that is why the DNA strand must be from 3 prime to 5 prime, because both are antiparallel. Okay, so now let's move to the next slide. Next one is the promoter. So what is promoter now? So promoter is the another component of the transcription unit. What is promoter? Promoter is the site of the DNA strand from which the transcription process begins. Yes? So now which part is called promoter? Simply, in the template strand, that means the strand from which the RNA is formed. That strand's direction is from 3 prime to 5 prime. Okay, that means the polarity, actually the polarity of that strand is 3 prime to 5 prime. So in this template strand, at the end of this 3 prime, the promoter is present. So what is promoter? Promoter is nothing but the nitrogenous bases of the DNA strand. Some specific nitrogenous bases are called as the promoter in DNA, and this promoter site is AT rich region. AT rich, here see, this promoter site is AT rich region means what? AT means what? A for adenine, T for thymine. That means this promoter site contains huge or abundant, a lot of adenine and thymine residue or thymine nitrogenous bases. Okay, so here see the promoter site. Okay, remember, it is located upstream of the structural gene. That means at the 5 prime end of the coding strand, which is the 3 prime end of the template strand. That means in this double stranded DNA molecule, in the template strand, the promoter site always present in the 3 prime end, and in the opposite strand, which is called as the coding strand, in the coding strand the promoter always occur in the 5 prime end. I will show you in the diagram later. Okay, so now this promoter site is rich in adenine and thymine nitrogenous bases, and that is why this region is known as the Tata box. Tata means what? Thymine adenine. Okay, so in prokaryotic cell or in prokaryotic transcription, this promoter site is called as the Pribnow box, which consists of 6 nitrogenous bases which are rich in thymine and adenine. In case of eukaryotic DNA, this promoter site is called as the Hogness box, which consists of 6 to 7 or simply 7 to 8 normally. Usually in eukaryotic DNA, the promoter contains 7 to 8 AT, adenine and thymine residue in the promoter site, and this promoter site in case of eukaryotic transcription is known as the Hogness box, and in case of prokaryotic transcription this promoter is called as the pre-now box. And what is promoter? Promoter is the site of DNA from which the production of RNA strand begins. Okay, so now let's move to the third component, which is called terminator, means ending. So the terminator is present at the 5' end of the template strand and the 3' end of the coding strand. Okay, at the 3' end of the coding strand which does not code for any RNA, which does not involve in the process of transcription. Only the template strand or antisense strand involved in the process of transcription. So in case of template strand, the terminator present at the 5' end. Okay, at the 5' end. Remember. So in this region, the process of transcription terminates or stops. Are you getting my point? In promoter, the transcription initiates. In terminator, the transcription process stops. That is why this is called terminator or termination site. Okay, so these three components are found in transcription unit. Okay, so now let's move to the next slide. So this is the whole structure of transcription unit. So here see, which one is called template strand? In the structure, see, this strand is called as the template strand. Yes, here see, this strand is called template strand, and whose polarity is from 3' to 5'. And see the opposite one. The opposite one is called as the non-template strand or simply the coding strand. Coding. The opposite of template is called non-template. Okay, so this is also called as the coding strand. So this coding strand does not involve in the production of RNA. Okay, so only the second strand whose polarity is from 3' to 5' direction, this is called template strand. Only from this strand the RNA is produced. Okay, so now here say, what is promoter? Which one is the region of the promoter and which one is the region of the terminator? Here see, this is the promoter. The dark green color region of the DNA molecule is called promoter, and this pink color region is called as the terminator. Okay, so this is the whole structure of transcription unit. That means this is just few sequences of the DNA molecule which take part in the transcription process. This is not the whole the DNA molecule. Okay, this is just one part of DNA molecule which involves in the transcription process, where this green part in this DNA molecule, the dark green part is promoter, which is present at the 3' end and of the template and 5' end of the non-template or coding strand, and the pink color is called terminator, which is found at the 5' end of the template and the 3' end of the coding strand or non-template strand. Okay, and always remember, from this template strand only the RNA is formed, and the direction of the RNA will be this way. That means from 5 to 3, because the template strand's direction is from 3' to 5', and the new strand will be from 5' to 3'. Okay, so this is the structure of transcription unit. And here see, what is upstream of gene? So upstream of gene means the 5' end of the coding strand. Okay, and downstream of gene meaning the 3' end of the coding strand. Coding strand, not the template strand. Okay, so the upstream of gene meaning the 5' end of the coding strand, downstream of gene meaning the 3' end of the coding strand, where the promoter is present and terminator is present. Okay, so here see, what is the function of promoter? So when the RNA polymerase binds to this promoter site, then the transcription process begins. Okay, then the RNA polymerase move in this direction. Okay, moves in this direction, and when the RNA polymerase reaches this terminator region of the DNA, then the transcription process or the synthesis of RNA strand terminates or stops. Okay, simply remember, in this transcription unit, the RNA synthesis starts from the promoter and RNA synthesis stops at the terminator. Okay, so this is the structure of transcription unit. Remember, transcription unit consists of three components: promoter, structural gene means this template strand is called as the structural gene, and the terminator. Okay, so these three are very important: promoter, terminator, and this structural gene, which is also called the template strand. Okay guys, are you getting my point? Okay, so this is all for today. So we will discuss on the transcription in prokaryotes in our next class. Okay, till then, take care and bye bye.